lang, the Group 7 First Reporter. So, our topic for tonight is all about the aspects of gene therapy. So, before we are going to our discussion, let's have an introduction first. We have an objective first. The first one is describe gene therapy and its various forms and assess the issues, potential benefits, and detriment to global health. The human body is composed of trillions of cells. Cells is the basic building blocks of all living things. The command center of each cell is called a nucleus, and it contains a chromosome. Chromosomes are made up of DNA, which is the body heredity material. So what do you think is the reason why the cells mentioned in this statement? Because gene and cells are interconnected to each other. The genes are inside almost in our body. Each gene contains instruction that tells to make a protein. So, ang protein ang ga-perform sa different tasks sa atong cell. Example sa atong eye color, powering muscles, and bacteria. And each cell has 23 pairs of chromosomes. And kato atong chromosomes contains proteins that help DNA exist in the proper form. Typically, every person has 2 copies of each gene, one of each parent. The genes are regions of DNA that code for proteins or contains instruction for making proteins, which are molecules that build, regulate, and maintain the body. So, ang every person daw has 2 copies of each parents. So, bantog, na example na isa ka bata ka nang magliwat sa mama, magliwat po siya sa papa. Or sometimes po, isara sa mama lang or sa papa lang. Every person has 20,000 genes and most are the same in all people. However, each person has a small number of genes that are slightly different. These slightly different in genes are why people have different features. So, what is gene therapy? So, a gene therapy is a technique that modifies a person's gene to treat or cure diseases. It is a technique that uses genetic materials, a piece of DNA, for a long-term treatment of genetic disorders. Another term of genetic is gene therapy techniques allows doctors to treat disorder by alternating person genetic makeup instead of using drugs or surgery. For example, if the person have symptoms of vision loss, but it can cure through gene therapy by injecting the lottery under retina and carrying the gene to replace the faulty one. Gene therapy has provided treatment for incurable diseases that previously has only temporary remedies. Some of these diseases are partly or fully caused by genetic mutations such as cardiovascular disease, neurodegenerative disorder such as Parkinson disease, vision disorders, blood clotting disorders such as hemophilia, and diabetes mellitus. So, now let's proceed in the basic process of gene therapy. The first one is replacing disease-causing gene with a healthy copy of gene. For example, if a doctor replaces the defective gene, may help treat certain diseases. The second one, inactivating disease-causing gene that is not functioning properly. For example, respiratory system of the baby is not functioning, but by activating the disease on it, the baby respiratory will function well. The last one is introducing a new modified gene into the body to help treat a disease. That's all for my report. So now let's proceed in the second reporting.
So, hello everyone. I am Laya Diana P. and I'm gonna report about the two types of gene. The first gene is somatic gene therapy, and the second is germline therapy. Somatic gene therapy involves a placement of a human gene into a living person's cell somatic gene that do not produce the sperms and eggs that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. So, soma means body. There are 200 types of somatic cells in our body, such as bones, muscles, and nerves. Somatic gene is a several essential functions in the development, maintaining, and regulation of an organism's body. Specifically in somatic cells, there are key functions of somatic gene: cell growth and division. Somatic genes regulate the orderly process of cell growth and division, ensuring that cells produce in a controlled manner to maintain tissue integrity and function. Second is differentiation, the functions of somatic gene therapy. This guides the specialization of cells into specific types which distinct functions. This process is crucial for the formation of tissues and organs with diverse cellular structures. Third function of somatic gene is cellular maintenance. Somatic genes contribute to the maintenance of cellular structures and functions, helping cells carry out their roles within tissues and organs throughout the organism's life. So the fourth somatic gene is apoptosis, programmed cell death. Some somatic cells play a role in programmed cell death, ensuring the removal of damaged or unnecessary cells. So the first function of somatic gene is DNA repair. Somatic genes are involved in DNA repair mechanism, helping to correct errors and damage that can occur during the normal life cycle of cells. So the sixth function of somatic gene is cellular signaling. Genes in somatic cells participate to signaling pathways that coordinate cellular activities, responding to internal signals and maintaining communication between cells and regulate various physiological processes. And the seventh function of somatic gene therapy is immune response. Certain somatic genes contribute the immune response, enabling the body to recognize to defend against foreign invaders such as pathogen and abdominal cells. Second is germline gene therapy. A germline gene therapy, DNA is inserted to the human reproductive cells, eggs, or sperm. In the human body, germline gene therapy will correct the genetic variants of reproductive cells of an individual, and it would be passed down to future generations. Germline, it occurs in the germline cells of a human body. Generally, this method is adapted to treat genetic disease causing variations to genes which are passed from the parents to their children. The process involves introducing a healthy DNA into the cells responsible for producing reproductive cells, eggs, or sperms. Germline gene therapy is not legal in many places. as the risks are outweighed and the rewards.
Good evening everyone, I am Narasha Asadi and the third reporter of Group 7. My report is all about stem cell gene therapy. Stem cells are the body's raw material cells from which all the other cells with specialized functions are generated. So there are three types of stem cells. One is embryonic stem cells. 2 is adult stem cells. 3 is indoled pluripotent cells. Stem cell gene therapy. Gene therapy involves the transfer of genetic materials into the appropriate cells. In genetic diseases, the stem cells of the afflicted tissue are often targeted. The adult stem cells of the tissue can replenish the specialized cells. Up is application of stem cell gene therapy. Cell therapy and gene therapy are overlapping fields of biomedical research and treatment. Both therapies aim to treat, prevent, or potentially cure diseases, and both approaches have the potential to alleviate the underlying cause of genetic diseases and acquired diseases. This is a therapeutic technology that enables the use of regeneration, including therapy for severe genetic diseases caused by gene deletion. So, advances of stem cell gene therapy. They can potentially replace cells and tissues that have been damaged or lost due to diseases, potentially treating conditions like spinal cord injuries, burn, and heart diseases, diabetes, and arthritis. So, the disadvantages of stem cell gene therapy. Genetic therapies hold promise to treat many diseases, but they are still new approaches to treatment and may have risk. Potential risk could include certain type of cancer, allergic reactions, or damage to organs or tissues if an injection is involved. Recent advances have made genetic therapies much safer. Next topic is the bioethics of gene therapy. So, gene therapy techniques allow doctors to treat a disorder by altering a person's genetic makeup instead of using drugs or surgery. Mechanism of gene therapy. A vector delivers the healing gene into a patient's target cell. The target cells develop into infected with the viral vector. The vector inheritable materials is fitted into the target cell. This is the example of viral vector. So that's all for my report, and thank you for listening everyone. Have a good night.